Well, good afternoon, everyone, and also a good evening, a good middle of the night, and a good morning for some of you. I know we have participants from all over the world, and we really appreciate your participation in the EV Journal Club. We've been doing this journal club since mid-March when things were shut down in the US due to the COVID-19 situation. And we've had a combination of journal club articles being presented and also some of people's own research. So this week, our topic is aging, senescence, and how EVs are involved in the process of aging. This is a paper that was recently published in the Journal of Extracellular Vesicles. This is the Journal of the International Society for EVs. And we have somebody today who's going to present this paper who is an expert on aging. So Nicole Noren Hooten is a scientist at the National Institute of Aging in the US. She's working just across town from where I'm located at Johns Hopkins. She's on a campus that we call the Bayview campus in Baltimore. And Nicole has made a lot of interesting contributions to the EV literature around aging and some other topics too. And so we're very pleased to have you here today, Nikki. And thank you so much for taking the time to share your expertise with us and also this interesting paper. And as sometimes happens on the EV Journal Club, we also have at least one of the authors with us today. So Emanuela Mensah, well, welcome to you too. Thank you for joining us today. I was wondering if you could perhaps just say a few words about this paper, the process of getting it published in JEV and where you are right now.
Okay, thanks Kenneth to introduce me, and good afternoon everybody. It is a pleasure for me to enjoy this meeting and to choose our papers for this journal club. I developed this work at Università Politecnica delle Marche, where I worked until last year under the guidance of Fabio Olivieri. This work has developed in more or less 4 years, and I need to thank all the collaborators, especially Michele Guescini at the University of Urbino, who isolated the extracellular vesicles, and the University of Bologna and London for the computational analysis. Now I started a new experience in London at Imperial College, and my PI is Luca Magnani, and I'm involved in the study of the chromatin landscape in oncogenesis, and in particular in cancer age-associated. And I surely will continue to work with extracellular vesicles, applying this knowledge in a different field as a cancer.
Excellent, excellent. Well, thank you for joining us again. And so what we're going to do here today is we'll have the presentation by Nikki. And during the presentation, everyone is welcome to use the chat function to make comments or to pose questions. So because of some security problems that we had a few weeks ago, we are not allowing people to unmute themselves, but Emanuela and some people from my lab and Nikki, they are all able to unmute people. And so we will try to compile the questions that we receive, and then we will give people the opportunity to ask those questions of Nikki and of Emanuela later on in the hour. So I will now hand it over to Nikki. Nikki, thank you.
All right, well, thank you, Ken, first for organizing the Journal Club. I know how much work it is organizing one that's monthly that we contribute to when we can see in person and now virtually. So I think we all appreciate your effort in continuing to move this field forward by organizing this Journal Club and allowing everyone to attend. I think we've all benefited from it. So I appreciate giving the opportunity to talk about this paper that you guys had mentioned, and I'm excited that Emanuela is on the call as well. Put some pressure off when the questions come in case I don't know it. Anyway, so this paper is the small extracellular vesicles deliver miR21 and miR217 as prosenescent effectors to endothelial cells. And so I thought I'd start off by giving everyone a background about cellular senescence, since this is not necessarily everyone's expertise. You know, I'm not going into as much extracellular vesicles, but more into cellular senescence for my background. Okay, cellular senescence is a state, a cellular state that's triggered by stressful insults such as oncogenes, DNA damage, oxidative stress, telomere erosion, and tissue culture. For example, if cells are proliferating in a tissue culture dish, continual passaging of the cells called replicative senescence will cause the cells to go into senescence. And senescence is characterized by a number of factors, including growth arrest, so irreversible cell cycle arrest. apoptotic resistance, higher levels of P16 and or P53, P21, higher senescence-associated beta-galactosidase activity, increased DNA damage response, telomere damage, DNA scars, DNA damage, including gamma H2AX, and higher levels of senescence-associated heterochromatin foci. Several years ago, Judy Campisi's lab coined the term SASP, which stands for senescence-associated secretory phenotype. So senescent cells secrete a large number of cytokines and inflammatory factors. So these are characteristics of senescence. So just like in the extracellular vesicle field, also struggled with identifying markers and being consistent with terminology, with also, you know, if you're characterizing senescent cells, they need to have these certain characteristics, not just one of the factors. So why do we care about senescent cells? Why are they important? So they regulate a number of different normal physiological functions, including they're important, senescent cells are important during embryogenesis, tissue reprogramming, tissue repair, especially wound healing, but also play a role in many pathological processes, including osteoporosis, glaucoma, neurodegeneration, and type 2 diabetes as shown here. It's known that senescent cells contribute to a number of age-related pathologies. Senescent cells increase with human age and age in mice, for example. In mice, selective elimination of senescent cells extends median lifespan and prevents or attenuates age-associated diseases. And so now there's this new class of drugs which are termed senolytics, and they selectively eliminate senescent cells and have been utilized in mice to prevent or treat age-associated diseases. And are currently, there's some clinical trials in people that using senolytics for age-associated diseases. There's also been a couple of editorials out there using that have posed the idea to use senolytics actually to treat COVID-19. So just to kind of give you a perspective, you know, when we age, we have senescent cells, but not all our cells are becoming senescent. So it's kind of the idea that you have this one bad apple here, but this bad apple can influence these other cells, for example, and that it's a bystander effect where the senescent cell can secrete SAS factors and potentially cargo in extracellular vesicles, as we will describe here, to influence cells in their neighborhood. So what about extracellular vesicles and senescence? So senescence is associated with increased EV concentrations. Now, sometimes some of these papers say increased secretion, but as we know, concentration is a balance from cells both by secretion and internalization. So I think sometimes the secretion term is overly used, and we just know that there's actually increased EV concentration. But nevertheless, senescence is associated with increased EV concentration in cancer cells, chondrocytes, dermal fibroblasts, and bone marrow stromal cells. And most of this work has really been very recently. EVs and vascular aging. So EVs from plasma of elderly individuals and from senescent cells, endothelial cells, promote vascular calcification. And there's reduced bone formation rate and reprogramming of monocytes towards a pro-inflammatory phenotype. So there's many epigenetic regulators of senescence, one main one being microRNAs. So I thought I'd give a little bit of background on them since they're a main focus of this manuscript. So microRNAs, just to remind everyone, are short non-coding RNAs. They typically negatively regulate gene expression by causing by the microRNAs binding to their mRNA targets and causing mRNA stability and affecting stability and translation. And there's been a lot of work on microRNAs in the context of senescence. So here's just a kind of view of microRNAs from one of my colleagues at NIA, Miriam Gorospe, in her laboratory, where you can find here MIR21 that's mentioned in this paper, and that's focus of this paper, and MIR217. So just to suffice to say that there's a lot of interest in microRNAs as regulators of senescence. And in addition, there's another epigenetic regulator that's discussed in this paper, methylation. In particular, so methylation is the addition of a methyl group in particular to a CpG island. It's an important mechanism to for stable gene repression. And they use terms, so I just want to kind of go over them to make sure everybody understands that global hypomethylation is the loss of a methyl group. And hypomethylation is typical in many different models of senescence and also in many types of cancer. So methylation is regulated by a number of different enzymes. one that they focus on in this paper is DNA methyltransferase 1, or DNMT1, and this is an important enzyme in maintaining methylation. And in addition, another regulator of senescence that they focus on, SIRT1. So SIRT1, in concert with DMT1, does help regulate and maintain methylation status. So SIRT1 is an NAD-dependent deacetylase. It regulates chromatin structure, genomic integrity, and DNA repair. It suppresses senescence and has a multitude of important biological roles. But it's also known, and maybe most famous, for regulating lifespan in model systems. So for example, the component in wine, resveratrol, that's known to extend lifespan in model systems, activate SIRT1. Here we start with the first figure from the paper. And again, what's important in the senescence fields is to carefully characterize your senescent cells. So they use as the model that's shown here, human umbilical vein endothelial cells, so HUVECs, which are an primary endothelial cells. And their model is replicative senescence, so inducing senescence by continually passaging of cells in vitro. So this causes growth arrest, which is observed here. These senescent cells have decreased telomere length. They are positive for senescence-associated beta-gal activity. So one of the things you can see, I tried to make the figures as big as I could possible, but there are a lot of panels, so I tried to do my best. But I'm used to seeing these images. So right here, I'm going to outline a cell. It's really large, it's flat, and there's this blue stain, which is quantified over here. So there's higher SA beta-gal activity. There's higher levels of P16, both at mRNA and protein levels, and P16 is a critical regulator of and marker in senescence. There's higher levels of gamma H2AX or phospho H2X, as shown here on our Western blot. Here's an inset, and here's the quantification. So they looked at SAS factors. So IL-1 beta, IL-6, and IL-8 are all higher in these senescent endothelial cells compared to control. They also, and I should mention, yes, so many of the key experiments were also performed with drug-induced senescence. So doxorubicin induced senescence in HUVECs, so that induces DNA damage in HUVECs. And then in addition, in another cell line, so human aortic endothelial cells, both replicated senescence and drug-induced senescence. And this is important because in the senescent field, it's important to make sure that the observations that you have are not just due to one type of senescence or in one cell line. So this is a lot of work, as Emanuela will tell you, to do this in the multiple cell lines, but it's really important in the field. So if we look at methylation status in the senescent and control HUVEC cells, there was no difference if you look at methylation in CpG islands in a gene, but there's a global loss of methylation, so hypomethylation in the intergenic areas, so areas that are CpG islands that are in shores or shells. And this is consistent with other data in the literature. And there's also a significant hypermethylation at non-CpG islands in a gene and not in a gene. So they were interested in global hypomethylation. And so again, the focus is on DNMT1, which is an important enzyme in maintaining methylation. So is this methyltransferase levels affected in senescence? So if they looked at the RNA, DMT1 is mRNA is lower, and protein level is lower in senescent cells, as shown here on the Western blot and quantified here. And they also looked at SIRT1. So SIRT1 mRNA is not affected, but the protein level is also much lower in senescent cells, shown here in the Western blot too. So is this due to different methylation status? So if they look at the methylation at the transcription start site, it's actually hypomethylated here in the purple box here compared to in senescent cells compared to control cells. And this is likewise in SIRT1 cells, for SIRT1 in these senescent cells where it is hypomethylated. So this suggested to the authors that post-transcriptional mechanisms may regulate DMT1 and SIRT1 levels during senescence. And so they asked, are DMT1 and SIRT1 levels regulated through an EV-dependent mechanism? So to isolate their extracellular vesicles from HUVEC culture media from control and senescent cells, they used a differential ultracentrifugation protocol where they've isolated large EVs from this 18,000 K pellet and then small EVs from 110,000 K pellet. So if they characterize, the cells and the extracellular vesicles from control and senescent endothelial cells. So here are some beautiful scanning electron images of control cells here, and here's a blow up image of it where you can see these cells. And senescent cells, you can see here that it's a very large stellate cell. And if you blow it up, there are a lot of little round things on the top that they presume are extracellular vesicles that are being secreted from these cells. And if the EVs are isolated, and then transmission electron microscopy images of small EVs or large EVs as shown here. So you can see that they're the typical size of extracellular vesicles. And then here's the large ones as shown here between control and senescent cells. So if you look at if they looked at the markers of senescent and or of these large EVs and small EVs. So the large EVs were positive for calnexin, and the small EVs were positive for the known small EV markers CD63 and TSG101. So they look at so they performed nanoparticle tracking analysis using a nanocyte. And so in yellow here is control, and purple here is senescent, and C are small extracellular vesicles, and D are large extracellular vesicles. And so if you look at the area under the curve and look at the concentration of the small EVs or large EVs, there's a significant increase in concentration of small EVs in senescent cells compared to control cells. But this difference was not observed when you looked at large EVs. There was no difference in size in the small EVs or large EVs comparing control and senescent cells, but there was a difference comparing the small EVs to the large EVs. So they also looked at transmission electron microscopy images for multivesicular bodies. And so here is one that's kind of outlined here, and there's an inset here showing an MVB. And so they counted all the MVBs in per micron squared in these images and found that there are significant higher numbers of MVPs in senescent cells compared to control cells. So these data so far suggest that there's a higher concentration of small EVs that are secreted from senescent cells. So what about the molecular cargo? As mentioned earlier, they're interested in microRNAs. So they perform small RNA-seq of control and from control and senescent cells, and also from small EVs and large EVs. Two microRNAs that were differentially expressed between control small EVs and senescent small EVs. 12 of these microRNAs were up and 10 were down. If they looked at the microRNAs that were differentially expressed between control large EVs and senescent large EVs, there were 23 microRNAs, 9 of them were up, 14 were down. And then here's the cellular content. So they're comparing control and senescent cells. There were 55 microRNAs that were differentially expressed, and then 14 were up and 41 were down. So this is consistent, being that the majority of microRNAs are downregulated. So this is consistent with data from other labs looking at microRNAs with senescence, and our laboratory and looking at microRNAs with human aging. I'll highlight it here, 2 of the microRNAs that they're interested in the paper, miR-217 and miR-21-5P, as these 2 microRNAs are differentially expressed both in the small EVs and in cells. And I'll talk about that a little bit more for this figure. So here's just showing a nice way to overlap the microRNAs that were detected. So 556 were detected. So if you look at the microRNAs that were differentially expressed in senescent cells, so here's the ones that were microRNAs from the cellular, here's the large EVs, and here's the small EVs. So they focused on the microRNAs, these 5 here, that were overlapping between, that were differentially expressed with senescence in both the small EVs and in the cells. So here's a real-time PCR analysis of miR-21-5P. So it was confirmed to be significantly upregulated in senescent cells compared to control cells, both in the cells and also in the small EV compartment derived from senescent cells. In addition, miR-217 was also confirmed to be significantly higher in senescent cells compared to control, and also in small EVs from senescent cells compared to control. So the levels of miR-21.5P and miR-217 were also confirmed in drug-induced senescence in HUVECs, and also replicative and drug-induced senescence in HAECs. And so here there are 5 microRNAs here that were of focus, but these 2 were the only one that were able to be confirmed by real-time PCR. miR-30A3P, 92A3P, and 130B3P were not confirmed by qPCR. So here's if they look at these microRNAs and whether they target these enzymes of interest, DMT1 and SIRT1. And so here, remember, in general, microRNAs negatively regulate their target of interest. So you would be looking for an upregulated microRNA to downregulate, since we know that DMT1 and SIRT1 levels are lower in senescent cells. So here they found that 21.5P can indirectly or directly regulate DMT1 and miR-217, and that's in the small EVs. So if the large EVs, there were none of the microRNAs that were upregulated or found to target these 2 enzymes. So interestingly, and they don't really discuss it too much, and maybe Emmanuel, you can chime on this later on, miR-21.5P is actually underexpressed in the large extracellular vesicles from senescent cells. And then if you look at the cells, there's a number of these microRNAs are known to target DMT1 and SIRT1. So again, they focused on miR-21.5P and 217, since they're both known to be regulators of DMT1 and SIRT1, and were differentially expressed between control and senescent cells and small EVs. So they looked at the methylation of the microRNA loci of the microRNAs that were differentially expressed in senescent versus control cells. So here's the methylation status of all these different microRNAs, and they found that there was, the methylation status was looked at 2,000 base pairs upstream and downstream of the transcription start site. And again, there were 55 microRNAs that were differentially methylated that they examined. So miR-21.5P was shown to be hypomethylated in senescent cells. And if the methylation of these microRNAs were imported to look at in a principal component analysis of the microRNAs, you can see a clear separation between the control, the methylation status of the microRNAs in the senescent cells versus control cells. And so about 50% showed senescence methylation changes in these microRNAs. And so they looked at whether, because you had proposed that if something was hypomethylated, then it would be upregulated. So this would suggest that methylation status is regulating this microRNA in senescent cells. So here, one of the top ones, again, is miR-21.5P, where it's shown to be hypomethylated and in higher levels. And it should be pointed out that there was no CPGs in the neighborhood of miR-217 on its locus on the EPIC chip, where they used analyzed methylation changes. So the question then is, do small EVs released from senescent cells modulate DMT1 and SIRT1 levels? So to do that, they first wanted to prove of concept that the extracellular vesicles from senescent cells are able to be internalized in control endothelial cells. So they labeled the extracellular vesicles with PKH67, and then incubated them with control cells for about 18 hours. And so here's immunofluorescence images showing the green dots reminiscent of fluorescently tagged extracellular vesicles compared to here's control cells incubated with PBS. And so to confirm that when these senescent extracellular vesicles are incubated with the control cells, can they transfer cargo? They loaded the extracellular vesicles using ExoFect kit from System Biosciences with a C. elegans microRNA. So this is C. elegans microRNA 39. And transfection of these small EVs with this microRNA, and then they added it to the control cells, they were able to detect using real-time PCR the C. elegans microRNA, suggesting that this is one, an efficient way to load a microRNA, and then two, it's able to be transferred into the cells. So if they collect EVs from control cells or senescent cells, incubate them with control cells, can they see differences in the two microRNAs that they found were in higher levels in senescent EVs? So they looked at first at miR21.5p. So there's two controls, just control cells untreated, or control cells plus control small EVs. So if the control cells were incubated with senescent small EVs, there was in these control cells there was higher levels of 21.5p shown with 217. So the control cells that were incubated with senescent small EVs had higher levels of miR217. And so in order to determine, is this due to the extracellular vesicles initiating a transcription factor, something that's causing higher levels of miR21 due to transcription, they looked at the primary transcript of miR21. And so a positive control was senescent cells. So they were able to detect this in senescent cells, but there weren't any increases in the primary transcript of miR21 with incubation of the senescent small EVs. So this suggests that the cargo, the increase of the miR21.5p and 217 is due to transcription, but due to potentially transferring of the cargo through extracellular vesicles. Okay, so do these senescent EVs change DMT1 and SIRT1 levels? So first I want you to focus right here on this panel. So if you have control cells and you incubate with senescent small EVs, there was lower levels of DMT1, and so there was reduction in protein levels of DMT1 and SIRT1. However, and these are, here there are two controls, either control cells or controls plus control small EVs. If they have these senescent small EVs and then they transfect in inhibitors of either miR21.5p or 217, so they did either in each individual inhibitor alone or in combination, they were able to rescue this effect. So here's higher levels of DMT1 and SIRT1 with a miR21.5p inhibitor and 217 inhibitor. When they add the two inhibitors alone, it's interesting because this is not a cumulative effect, and that's maybe something we can talk about later in the discussion. But nevertheless, there still is a difference if you compare here to here. And here's quantification of those Western blots. It's a busy graph, but I'll just kind of walk you through a little bit of it just to highlight that they saw a significant decrease in SIRT1 levels with senescent EVs, and this is rescued with either of the inhibitors or the combination. And here's, if you look at DMT1, reduced levels with senescent small EVs, and this was rescued partially, not to the extent of SIRT1, I guess, with the inhibitors. So senescent small EVs reduce DMT1 and SIRT1 levels. So what about other cellular functions? Does it affect proliferation or senescent markers? So first they looked at cell proliferation. So they did this using a Ki-67 immunofluorescence. So you can see these green dots here show proliferating cells in control conditions. Control cells plus small senescent small EVs, you see reduced Ki-67 staining. And then if the extracellular vesicles are transfected with miR21.5p or 217, this rescues that effect. And this is quantified here on the right, where looking at Ki-67 percentage stained cells, where senescent EVs reduce proliferation, and this is rescued by the inhibitors. They also looked at an additional proliferation marker, PCNA. This is mRNA levels, so it was reduced with senescent small EVs, and this was rescued, at least in part, with the inhibitors. So they also looked at Hoist staining here and looked at the coefficient of variance of Hoist staining. This is a slightly unusual way, I think, to look at. So they're looking at senescence associated heterochromatin foci. And so you can see here that there's these DAPI or Hoist staining, it's kind of punctate more. And so they looked at the coefficient of variance of this. And maybe Emanuela can mention a little bit about why they chose this technique. It's sometimes difficult to quantify. but there was a significant increase with senescent small EVs, and this was perhaps not as robustly rescued by the inhibitors. In addition, they looked at several cyclins, including cyclin D1, cyclin A1, and cyclin B1 levels, looking at the RNA levels. So senescent small EVs reduced cyclin, and this was rescued by 21,5P, but not 217. So that's, I think, also interesting. And this was also observed with senescent, with cyclin A1, that it was rescued. So senescent small EVs reduced cyclin A1 and cyclin B levels, and it was rescued by miR-21,5P inhibitor, but not miR-217 inhibitor. So what about some of the senescent phenotypes in these cells? So again, they had control cells that they incubated either they either had untreated or treated with senescent small EVs. And here they looked at apoptosis markers. So they found that SN5 was higher in senescent control cells that were incubated with senescent small EVs. I think this is interesting because typically senescent cells are actually resistant to apoptosis. And so, you know, I find this interesting, and I wonder whether if the experiment, so this was done in 18 hours, so a shorter time course. So I wonder whether, you know, what would happen if this experiment was taken out longer, and to see actually if the cells go towards senescence or do they go towards apoptosis, because those are typically two different pathways. But nevertheless, there's senescent markers that are upregulated. So if you look, we look at P16 mRNA levels, they were higher in control cells that were incubated with small EVs. And if you look at IL-6, IL-8, they were both significantly increased. So SAS factors were higher. There wasn't any difference in IL-1 beta. So there were many different senescent markers that were upregulated in control cells that were incubated with senescent EVs. Now, if you look at methylation of the miR-21 locus here, and so this is in control cells or senescent cells or control cells plus senescent small EVs, there's hypomethylation of this of the miR-21 locus. So this suggests that there's reduced methylation of miR-21 locus when senescent small EVs were incubated with control cells. So next they wanted to look at the plasma EV levels of various these microRNAs with human age. And so they have either young individuals who are around 50, elderly individuals, which were 74 mean age, and centenarians, which were 102. And there were 4 individuals per group. And so they isolated EVs by QEV original column from ISOM. And so they had detected 1,186 microRNAs, and 360 were present in EVs in at least 2 of the subjects and were used for analysis. So they did a principal component analysis where you can see clear separation of the microRNA expression levels from young individuals that were less than 65. Here's the older individuals, and here's the centenarians. And it should be mentioned that miR-217 was not detected in plasma EVs. So if they looked at expression of miR-21,5P in these extracellular vesicles, there's what they describe as a U-shaped expression pattern where microRNA 21,5P is lower levels in younger individuals that were below 65, where higher levels were shown with human age, so in the elderly, and this was in reduced levels in centenarians. So again, like this U-shaped response, U-shaped levels, which is also similar to what they've observed previously in looking at miR-21,5P levels in whole plasma, so not separating out extracellular vesicles. So if these microRNAs were then imported into Ingenuity pathway analysis to look at the diseases and disorders, one of the top pathways was inflammation of organ, which is consistent because Claudio Franceschi, one of the authors on the paper, is coined the phrase inflammaging. And so inflammaging is a state of chronic inflammation that's observed in the elderly. And so here in this IPA analysis, they found that TNF alpha or TNF and TP53 are major hubs in this inflammation of organ top disease and disorder. And so here's the model that's proposed from the experiments in this study, where if you have senescent cells, they secrete both small EVs and large EVs. And these small EVs contain miR21-5P, which is in this light green, and miR217 in this dark green. And then these senescent cells have the miR21 locus is demethylated. And so this demethylation of miR21 causes higher levels of miR21-5P and 217. And this could be due to lower levels of DMT1 and SIRT1. Now, if you have these senescent cells and they're secreting these small extracellular vesicles, it could cause this bystander effect, where they're affecting cells in their neighborhood by secreting these extracellular vesicles. And so they're being taken up by control cells. And this could be propagating the effects of senescence by lowering DMT1 cells, DMT1 levels and SIRT1 levels, and proliferation markers like Ki67 that was measured in PCNA, and cyclins, including cyclin D1, but that there's increase in apoptosis markers and also other senescent markers like P16, IL-6, and IL-8. And that some of these effects could be blocked with microRNA inhibitors, as shown here. And then there also suggests that miR21-5P as a biomarker of senescence inflammaging, such that individuals that are young have lower levels of 21-5P. And again, this U-shaped curve where there's higher levels in individuals that are elderly, over 65. However, if you look at centenarians, they're known to be resilient. They have lower levels that are similar to actually the young. And so I think this points to, they're pointing to circulating microRNAs as biomarkers of senescence. So I wanted to point out, so we did a meta-study a couple of years ago looking at circulating microRNAs that change with human age. And so there's one of the problems I think in this field is that there's several different studies that look at these microRNAs, and some of them may differ if they're up or down in humans. And so I wanted to kind of point out that 21-5P, and this review is now 4 years old, so there may be other studies, but there was several studies that looked at it, and it differed in that 2 of the studies it was up with age, and 1 of the studies it was lower in age. But some of this is some of the problems in the aging field that we find is that people define young and old differently. So that may be one of the reasons that this microRNA was decreased in 1 study and found to be increased in 2 studies. And so something to just keep in mind. And then in addition, I also wanted to point out that this group published an editorial in JEB in 2019 entitled Extracellular Vesicles Circulating in Young Organisms Promote Healthy Longevity. And so I wanted to point out this figure from the paper, because ironically, I presented one paper that it discusses in this journal club a couple of months ago. And this paper was a paper by Yoshida et al. In Cell Reports, looking at extracellular nicotinamide phosphoribotransferase, or ENAMPT. And it's found that this enzyme is in extracellular vesicles, but it's actually decreases with age. So this enzyme regulates NAD levels, cellular levels. And so they propose in this editorial that reduced ENAMT and then higher levels of senescence-associated microRNAs are important for aging. And that, what is shown in this paper, but and what they're also proposing is that if you have young mice here, and you take blood, plasma, isolate the extracellular vesicles, they have high ENAMT, lower senescence-associated microRNAs. But then if you are able to inject these into older mice, it may lead to higher lifespan, increased lifespan, health, and physical activity. So with that, I'll stop and be happy to answer questions. And yeah, thank you. Thank you for listening and attending.
Great. Well, thanks so much, Nikki.
Thanks.
I enjoyed your presentation. And we've had a few questions that have come in on the chat box. But I thought maybe we would start with some of the issues that you raised, some of the questions that you had for Emanuela, who, yes, is also unmuted now. So you had asked about the Herxt staining and why the Herxt stain was used, and then also about the apoptotic versus senescent cells, and whether those are mutually exclusive, and whether a different time course might have had some more information to reveal. So do you want to start with those, Nikki?
Sorry, Kenneth, could you repeat, please, because I don't have these questions.
Sure. So, Nikki, could you bring up the point that you had about the DNA stain?
Yeah, so one of the questions I had was, so when you did the Hoist staining and you quantified senescence-associated heterochromatin foci using coefficient of variance of that. Yeah. That was just a slightly unusual way that I hadn't really seen before. Yeah. So I was just wondering.
Yes, clear. It was difficult to analyze this with this technique, but we found this technique in a paper, in a different paper. So we did these experiments on base of this paper.
Okay, all right, thanks. The other question was that you saw a higher level or increase in annexin 5 staining or in the control cells that were incubated with senescent small EVs, but typically, senescent cells are resistant to apoptosis. So I was just wondering about that.
Yes, we detected the young cells with the vesicles released from senescent cells, basically with the exosomes, the small vesicles of senescent cells, and we analyzed the effect of these small vesicles in the old cells, in the young cells, sorry.
Yeah, did you ever take out the experiment longer to see whether the cells underwent apoptosis or whether they continued on the senescent?
We treated the young cells with the small vesicles released from senescent cells for 18 hours.
Okay. Okay.
Because after 18 hours, the cells are changed their morphology. So the HUVECs are are cells that require very, very, very cure. So if you take these cells and culture these cells for a time more than 18 hours, you observe the different morphology of these cells. So to avoid this problem, we choose this time to perform all the experiments.
Okay. All right. Ken, are there other cells or other questions?
There are always other cells.
Exactly. Yes.
We have many cells, and so I guess yes, we do have some other questions. So one is from actually we have two questions from Amrita. So Amrita, I have just unmuted you, and you had some questions about the microRNAs, but then also about some other factors. So
yes. Yes, can you hear me?
Yes.
Yes.
Okay, great. Yes, so I have two questions. So the first one is that, you chose two microRNAs that are expressed in the small EVs as well as the cells. Yes. Would it be, I'm sure that it's going to be helpful that if you actually look at, the microRNAs that were expressed uniquely in the small EVs, because I feel like probably you might, of course, you are able to show a mechanism of action through, the miR21 and miR217. But if you look at the uniquely expressed cargo in the small EVs, that will also be beneficial. So I just wanted your thoughts on that, on what basis you chose these two microRNAs.
Yes, we choose these two microRNAs because are expressed both in cells and small extracellular vesicles. And we didn't test the large extracellular vesicles because we observed that the amount of vesicles released between young and senescent cells of large vesicles didn't change. And so we choose to study only the microRNAs in small and cells.
Okay. Yeah, and I guess you kind of answered my second question, which was that did you also test the effect of the large EVs? Because, if they don't have these two
Yes.
microRNAs and don't see the effect, then that would also be proof of concept that this effect is specific to the transfer of these microRNAs through the small EVs.
It will be interesting, but for the moment, for this paper, we focus our attention only on the microRNAs in common in cells and the small extracellular vesicles. And why? Because the large vesicles, the amount of large vesicles between young and senescent cells didn't change.
Okay. Okay. Thank you.
Great.
Welcome.
Great. So, I think that very closely related to Amrita's first question was one by Brian Bird. So Brian, I've unmuted you. Do you want to ask us about the interpretation of the exclusivity?
Thank you. Can you hear me okay?
Yes, we hear you.
Hi, thanks, Ken. Thanks, Nikki. And this is really very interesting paper, a really nice body of experiments. What I was wondering is that the authors chose a very fruitful approach by focusing on the microRNAs that were differentially regulated both in the small EVs and the cells. And it was not immediately obvious to me why that would be the subset of microRNAs that would be of interest. Is there some intuition about the physiological significance of the microRNAs that are regulated, I guess, similarly in the EVs and the cells versus ones that are, for example, different small EVs versus the ones, but not in cells, I should say.
Emmanuel, do you want to answer, or do you want me to answer?
Okay. Yes. Sorry.
I don't know.
Sorry, I had some problem with the connection.
Okay.
And so, okay, I'm here. And yes, we choose these microRNAs because the selection is because the smallest cellular vesicles increase in senescent cells compared to younger ones, the release of small extracellular vesicles. Then when we analyze the microRNA expressed in vesicles, we focus our attention only on two microRNAs because there were 5 microRNAs in common between cells and small extracellular vesicles. And among these 5 microRNAs, only 2, so the microRNAs 21 and 217, are more expressed in senescent cells compared to younger ones in a real-time PCR. So the data obtained in small RNA-seq compared to real-time PCR demonstrated that only the MIR21 and the MIR217 have the same expression in so increase in senescent cells. So the selection is due for this reason.
And Brian, just to kind of step off that, Emmanuel's response, it is known that there is a motif, in some microRNAs that actually preferentially sorts them into vesicles, an exomotif that's known, and they can bind to certain RNA binding proteins that may help them transport into extracellular vesicles. I don't know if these 2 microRNAs fall into that category or not, having that exomotif, being that they're exo, or if it's just the nature of these cells are producing more, so more goes into the extracellular vesicles, at least the small EVs, because there's more being expressed in the cells.
Yes, this is the problem, yes.
Yeah, and I think it's important too to just note, for the sake of interpretation, that if we do find microRNAs that are only in the vesicles and not in the cells that they came from, how exactly do we interpret that? I think it's important not to necessarily think that they were never in the cells, because
like a waste for the cells.
Right? Yeah. So they had to have been there at some point, and maybe it's just simply a matter of them being at very low levels and having a very highly selective export into the EVs. So just a matter of interpretation there. Good. So we have a few other questions that have come in. I had one from somebody who has a microphone problem. So Yvonne had asked about whether it's possible to distinguish between the microRNAs that might be associated with aging versus diseases of aging. And that's something that would probably require, a larger cohort study. So I was just wondering if you have something to say about that. And I also wanted to give Pablo, so Pablo Munoz, I think was, let me see if you're unmuted here.
Hello?
Pablo? Yes, go ahead. Why don't you go ahead and ask your question too, because it's really.
Yeah, thanks. This is very nice. I am very new to the extracellular vesicle field. I just want to learn, and I got my attention about the difference between this microRNA in the literature. The Nicole said that sometimes they are high and they're down. So I was wondering if, in particular, this paper is related to microRNA with inflammation. I wonder if there is an impact of the diet of the subject they are studying, if a non-Western diet would show different extracellular vesicle or different effect to a subject that has a Western diet, for example. Since the non-Western diet high in antioxidants and omega-3 and omega-6 can affect the anti-inflammatory of all.
Maybe yes, because the inflammation is related by environmental factors such as diet. So maybe yes.
Okay.
Yeah.
So I think those type of studies are always something that people are interested in, especially given, for example, in mice, at our institute and many other institutes are very interested in diet and compositions of diets. But I think in general, those type of randomized controlled trials or observational trials in people aren't necessarily always done to compare, for example, microRNAs. I don't think that there's been one to look at the microRNA profiles there. And to kind of piggyback on the first question, people are all, we've studied microRNAs with aging, in Emmanuel's, this paper, and they've had other papers from this group looking at different microRNAs with aging. And so to try to separate from age-related disease, we try to do the best we can. As far as, I know in this paper, they excluded individuals with diabetes, cancer, and several other exclusion to try to separate aging from age-related disease. But sometimes that's difficult to do in aging studies. But there are, a lot of papers. We've had several papers looking at microRNAs and extracellular RNAs with age. So they do change, and it's different, and it's interesting. And I think it's something that I know the Extracellular RNA Consortium is interested in. Although I think in general, many of the different projects have focused more on disease than aging or in normal populations. So I think that's something that we can definitely expand upon.
Yeah, thank you.
Great. Okay, we have a question now from someone named U0107156. I'm trying to unmute this this guest of ours, and I can't seem to do it. Oh, cannot speak out loud right now. Okay, good. Okay, I will ask the question then. So U0 asks, based on these results, what might you recommend for decreasing senescence in primary cell culture? So this is kind of like, what does this mean practically? Can you stop senescence using these findings?
Ooh, big question. I mean, I think, I think if you take some of the figures from here, one of the, factors that affects aging is physical activity. So that is, when people always ask me, how do you stop aging? Well, first they always ask me about their wrinkles, which I don't really work on that. But one of the known factors that affects, health span and lifespan in mice is physical activity. One of the, and it's a really hot topic right now about senolytics and xenomorphics, and there's, on using these to treat age-related diseases. And there's really promising clinical trials, and also, but they're starting in some of the, because most of these drugs that are senolytics are actually already FDA approved, and they've been around for a while, and it just so happens that they selectively kill senescent cells. And so there's a lot of promise right now in using these drugs for various or age-related diseases, and many that don't really have many current therapies or therapies that are that effective.
Anything to add, Emanuela?
No, nothing to add. Perfect.
Okay. All right. All right. And our questioner has said, so this was actually from Lucas Treps from Leuven. So thanks for joining, Lucas. Next question was from Genevieve about the blood vessels. Why these cells?
I'm kind of asking a bit more what happens at the level of the organism. So if you look at the effect of the EV secreted by endothelial cells, do you expect that senescence is being transported to organs, or is it really specifically a problem with blood vessels?
Well, I mean, I think one thing I think that's a big question in the field in general is, you know, if you have senescent cells, what effects are local, like affecting their neighborhood, and what effects are systemic? I mean, it is known, Luigi Ferrucci, you know, at our institute has found, you know, and Claudia Francesca know, IL-6, for example, is an important inflammatory marker that increases with age, and there's inflammaging, but you know, whether those are contributed from senescent cells is not known, if that makes sense. And so I think some of the effects, we know that there's definitely, you know, in a lot of the mouse models where, you know, senescent cells are eliminated or there's senolytics, and some of the tissue specific effects where senescent cells are removed in the brain, for example, or in various organs that it has beneficial age-related effects on that specific disease. But I think we're still understanding, and maybe Emanuela, you can mark on or talk about the choice of the endothelial cells and HUVECs.
Yes, we choose the endothelial cells because they are an example of replicative senescence, and the endothelial cells are a perfect example to aging, because the endothelial cells are going to replicative senescence, and we can minimize what happened during the aging in an organism. So we choose this kind of cells. And yes, I think that the effect of senescent cells is not restricted to a single cells, but influence the neighboring cells. So one young cells nearby the one senescent cells can become senescent as well.
Thank you.
Great. Next question is from Joanna. Joanna, do you want to ask your question about the stressed cells? Maybe having some problems with the microphone. In any case, the question was, would it be interesting to test microRNAs that are coming from stressed cells to observe their effects? Is this an experiment that you've
yes, it will be interesting. Yes, it will be interesting studying the effect of the vesicles on stressed cells. Yes, sure.
Good. And then we have one last question, and it is from Ellie Tedford about the central nervous system.
Hi, yeah. Very nice talk, thanks. Have you or will you maybe investigate the effects of these EVs on other cell types, like neurons and glia? So I was just thinking AD or dementias, there've been quite some studies about using young blood as a treatment. They do these parabiosis experiments where they effectively sew an old and a young mice together. So they share a vasculature and they see huge cognition improvements in the old mice.
Thanks for these questions. For the moment, no. We analyzed these things in the HUVECs and in replicative and in inducted senescence. Not in the central nervous system, but maybe thanks for the suggestion. It will be interesting in the future.
So there were two really prominent papers that came out last year, one from Darren Baker's lab at the Mayo Clinic and one from Mark Mattson and Miriam Gorosky's lab at our institute, looking at senescent cells in the brain. And they use different models, and so a tau model and also an A beta model to look at the contribution of senescent cells, in particular either oligodendrocytes, progenitor cells, or astrocytes to neurodegeneration. And so eliminating senescent cells, either the astrocytes or the OPCs, had a beneficial effect on the mouse models. And so this is something that's, I think, very active in the field. And I know Tony Weiss-Corey is really, has some really nice data looking at the parabiosis and the young blood versus the old blood. And I think, you know, the one example is this paper here where, so in this paper, they have old mice where they've injected young extracellular vesicles and see a beneficial effect on lifespan. And that's attributed as far as lifespan to EVs. Now that experiment, they just looked at lifespan, and even actually they ended the experiment and some of the mice were still living and they were pretty old. So it showed that some of the beneficial effects of, for example, that they're seeing in the parabiosis experiments, maybe through extracellular vesicles. So I think that's something that we'll see in the future, and it will be, you know, very interesting.
Excellent. Well, that brings us to the end of our hour. So I'd like to thank our presenter, Nikki Noren Hooten. Thanks also to Emanuela Mensat for joining us and answering some questions, and she was the first author of this publication. And thank you all for joining. Thank you to the.
Thank you again.
Yeah, thank you, especially to those of you who have volunteered to give some future journal clubs. So if you have not done so, feel free to reach out to me. I have about 5 or 6 people that I still need to respond to. So I really appreciate the outreach there. So thanks everyone. Take care, stay safe, and we'll see you next week.
All right, thank you.
Thank you very much again.
And thank you,
Bye-bye.
Emanuela.
Stay safe. Bye-bye.
Bye-bye.